Okay, so hello everyone. Welcome to our 12th session of Computational Bioengineering. So today we are going to look into computer vision for microscopy. There has been some rearrangement, as you might be aware of since last time. So we have rescheduled. So yeah, we have 3 more sessions. These are classical sessions, the computer vision today for microscopy. The next one will be about bioproduction. Will be given by Mariapia. And then we have a live from the lab session, and we will keep you in touch about this, how we are going to organize this. So especially for our online audience, I think for the local audience here, we will go to the lab. And perhaps for the online audience, we will perhaps do some cuts together, from nice protocols. We keep you in touch how we do this. Some other announcement. There is a new homework here. So this will be based on the AI sessions, the last two AI sessions we had, and you're going to give it a read. It's very nice problems of mechanistic versus data-driven classifications. So hope you have fun with this. And so let's get started with this course. So today we are going to learn about microscopy. And of course, we will learn from it from a computational perspective. So that's microscopy, right? You might remember the measurement sessions. And in the measurement sessions, what we did is you need to observe your experiments, whether they fulfill your hypothesis that you made. And in the measurement section, we looked at bulk measurements, right? So we had some sample with bacteria in it, and we measured some properties like optical density for, let's say, a certain wavelength and so on, or a Raman spectrum, right? But these were properties of the complete complete sample of the population level. So let's say this is a population level. Yeah? And sometimes we want to take a closer look. We want to know what is the behavior of each individual cell. Yeah? So this will be cell level measurements. So we could have equally called this cell level measurements, this lecture. Yeah? And what we are going to have is typically images that we did with a microscope. And you will see some bacteria cell. For example, here's a bacterial cell. And there's another bacterial cell in here. Yeah? And the things you might observe, you might be interested in is how are they going to move? For example, what is the movement of this? Or you might want to say, how many cells are in my picture? Right? This is a very precise count because it's really a natural number, a small natural number, compared to, for example, the rough OD measurements here. Or you could be interested, even in interactions, they could interact, right? This could here interact with this one, for example, by bacterial conjugation. Or you could be interested in duplication rates, for example, this duplicating and being suddenly 2 cells, right? And you can, once you have such an image, so this is an image, right? And we are going to look into what is actually an image in a second. You can create several statistics from this. Yeah? Like statistics, perhaps being here quoted, because what we are often interested in is kind of abstract properties from these images, right? So let's say you have many images from your experiment. So this can be images that are kind of not connected, or they can be images from a timeline, from a process that you took off. Yeah? So the sequence has actually a meaning in this case, yeah? For example, to track movement. Yeah? And you want to kind of abstract this. You want to get the essentials from these images. Yeah? And of course, what is an essential depends a lot on your research question. Yeah? So someone might be interested in just the number of cells in these images. That's a kind of an abstract property. Yeah? Someone else might be interested in where exactly are the cells and so on. Yeah? And of course, we cannot in this lecture look in all possible research questions that you might have with such images. But what we can do is actually, we can discuss common questions that you might have. And a very common question, a very common question is actually, let's say you have here your image, and let's look for the moment at a single image. Right, you might be interested in a problem that is called segmentation of this image. Right? So perhaps the even more abstract problem would be labeling this image. For example, you could label this image with there is cells on it, or there are E. coli on it, right? This would be a label, but it doesn't tell you where and things. And the next concrete thing is segmentation. So segmentation can come in often in two aspects. Either it puts a bounding box around this and tells you there is an E. coli inside here, or it's even pixel level segmentation, where it really gives you this pixelated area here and tells you this is an E. coli. This is a pixel that belongs to an E. coli. And so this is for single images. Segmentation also comes in two small aspects, and we will actually look at both ones. So you could, for example, in the pixel level segmentation, and we are going to look into pixel level segmentation, you could label this and this pixels here with the same label. So you could just say this pixel and this pixel belongs to a cell, and that's it. Or you could differently label individual cells. So this could be labeled, for example, with the value 1, and this could be labeled with the value 2. So you know that this pixel here belongs to a different cell than this pixel. And we are going to look into both ones. So for some aspects, you don't care to the individual cells here, and sometimes you care about the individual cells. And there is an even more perhaps advanced problem, but we are not going to cover this in this lecture. This is actually tracking. So tracking would be you have here the next image, and this is from a timeline T, right? And let's say you have here another, or the same cell, and this would be again 1, and this has now moved, let's say, here, and this is again 2. Right? So you kind of follow the trajectory of the individual cells, and you know that this cell here belongs to this cell and this cell here belongs to this one, and not vice versa, for example. Of course, you can imagine with cells it's a bit complicated because they can go out of view. They can just disappear here. New cells can come into view. They can duplicate. So what do you do then? Just be careful, or they can just die, dissolve. So you have to be very careful, of course, with this. Okay, I think this is the setup we are going to look into. And perhaps we will first discuss with a very simple question. And this will be, what is an image? In particular, every one of you knows what is an image, but what is an image in Python? And these are just some very basics, but we want to make sure that everyone is on the same page. So an image is essentially a function, right, that maps a pixel to a color, right? And in the particular case of the images we are looking into, this is like 2D images. Like we do not have to address this pixel explicitly by encoding the complete function, but we can make sure that, for example, we are traversing the image from the top left to the bottom right, and we just append in a list, or like in a structured list, it will be finally of multi-dimension, but it will be a sort of a list. We just append one color after the other, by fixing the traversal. And this is actually what is done in, for example, in NumPy. So an image would be, you can very simply create an image. For example, this would be a 4x4 pixel image. And we are just creating one with zeros. Yeah? We are telling it the type, so the type of each pixel will be an integer 64-bit. And then we are setting the top left, so the 0, 0, we are setting it to the value 1, and everything else is kept as 0. So this is an image, right? And here we're using the convention that we are traversing this pixel. So we don't need to explicitly write we are mapping pixel 0, 0 to 1, we are 0, 1 to 0, and so on, but we're just traversing this, and this is why you see this 2D structure. The 2D structure, I mean, we could completely flatten this list also, of course, but then we lose the information like how wide this image is. I mean, if we have, of course, the information 4, 4, then we could present it just as a list, but here this is already in this data structure here. So this is actually really the image, how you will see it as the lines, right? And with the usual libraries that you like about plotting, for example, matplotlib, you can view such an image. So there would be, this is how you can view an image. You just call imshow, and it shows you this image here. I mean, here we have axes. Of course, we don't need that, for example, for this image, but this is just, and you see here the addresses of the pixels. So we have, for example, the first position would be the 0, and the second would be the 0. So we set this to white, and everything else is black. And one thing to say here, why is this white? So how do we know that, for example, 1 corresponds to white? And this is actually not fixed. This might depend very much on your color map. But here, in this case, we're showing the color map gray. And here it is defined that everything is between 0 and 1. And 1 is mapped to completely white, and 0 is mapped to completely dark, so black. And everything else would be a shade of gray. Just to see that I was not things making up. Like if you put another color map, for example, chat is a very commonly used color map that we will be using also for labels. The image looks completely different, right? So this is underspecified. In a way, if you don't specify your color map, it's not clear how it visually will look like. I mean, the array is the same, of course, but the visualization is different here. So that was for the simplest images where our color is just a number. So in our case, it was, for example, a number between 0 and 1, a float. Or it could have been also an integer. I mean, we didn't make use of it that it's a float here. But you kind of can have more complex colors. For example, this allows you kind of grayscale images to be represented, even if they are not shown in grayscale, but they might be shown in different color maps, but they have only a one-dimensional color, which is a grayscale image. You could have multi-dimensional colors, and most commonly used is, for example, a three-dimensional color. And this is typically so here we are creating, again, something with a certain height, something with a certain width, so again 4 times 4, But now we are what is called creating 3 channels. So we could have viewed the other image as a 1-channel image. And in this last channel, this encodes actually for a triple, and this triple here would be an RGB encoding, where the colors are typically encoded between 0 and 255. But again, this depends a lot on where you have your data from. So you cannot assume that your triple color is between 0 and 255. For example, you might even have values that are 16-bit values, or many cameras give you 10-bit values here. So it really depends on your application and what on your image source. So the question is, can you have even more than 3 channels for a color? Do you have an idea? Like for settings where you have even more than 3 channels than RGB? Yeah, you could have saturation information. Typically, what is saturation information is, it just levels off. For example, if it would just say 255 if it's saturated, yeah? In this particular case. But for example, you can think of something like fluorescence. Yeah? Not a normal camera, but a microscopic camera might give you fluorescence for yellow, fluorescence for red, and so on and so on. You can have an arbitrary number of channels that are not shown in classical images, but in microscopic image it would make sense. Completely, yeah. Any kind of spectrum you can think of, you can put there, yeah, completely, yeah. Mm-hmm. Very good, yeah. We will not use that in this course, yeah. We're using just classical light microscopy here, but you could completely do that here also. And yeah, if we plot that, this is how it looks like. And this is now following the RGB code, right? Like we have everywhere black pixel, and here we have a purely red pixel. Yeah? So this is the classical. Yeah, just some words about bit depth. You have to be very careful about this. This is not a big thing. Yeah? It's actually, I mean, it comes normally with the application, with the library, and so on. You know the bit width, but I mention it here because it's really a source of painful errors. Yeah? and when doing the preparation of this lecture, like of course there were bit errors and so on. You have to be very careful if you're using a U in 16 or a U in 32 or whatever. Your training might go completely wrong. Your images might show completely black and nothing, and so on. Like it's a painful source of errors. So always check your bit depth, yeah, and what you're doing here, what's expecting, and of course what is particular about it is like we show you in this course, your build chain, your image chain, yeah, has of course many software from authors that are not at all related. Yeah? So something might need this in this bit width and this in the next one. So there is a lot of transforming images between the different stages. Yeah? And this is typical errors. Yeah? So perhaps the general rule is like always sit in between the stages and check. You will see in our code, like we put a lot of assertions for the dimensionality and so on and so on. And this is exactly for such reasons. Same painful thing, normalizations. Yeah, many image stages require something between 0 and 1 or between 0 and 99 for whatever reason, 0 to 25 for whatever reason, and so on and so on. Be super careful about this. Last very typical source of bugs is the channel ordering. You have seen, perhaps for grayscale images. Here we will look a lot at grayscale images for segmentation. You have seen, like we started a grayscale image with a two-dimensional image. Yeah, so we just had the height and the width, and that's it. Yeah? Here you see suddenly there is a channel, right? And for RGB images we need a channel, of course, but for grayscale it's not sure. Yeah? And some libraries require here that this is actually a pseudo dimension. Is just 1. Yeah, so this is again some painful thing, and that's not even enough. For example, libraries that NumPy, PIL, OpenCV, and so on, they prefer this format that we showed you just before, that we had in. If you go to PyTorch tensors, they almost always want this format here. So you have to do transposings between them all the time. Yeah, so also super painful source. And here we show you how to create such a tensor and how to transpose, for example, from this to this format. It's simple. I mean, it's super simple, but it's just something you can easily forget there. And that's a little reminder here. Okay. So that was our little primer about images in Python. Now we can start with the real problem, segmentation. Yeah, so I told you already there is 2 types of masks, yeah, and perhaps we have to talk about what is a mask. Yeah? So you can think of a mask very simply as this is an image, and that has some, for example, some object, right? It has, for example, this object here another object, yeah. And then you might have something which is this is the image, and then you might have something that is a mask, right? And this mask is often encoded in the way that each pixel has the value 0. So the 0 means actually background. Yeah? And then where there is an object, for example, in the segmentation operam here, you would have this here. You have, let's say, ones, right? So in this case, I don't know, let's say we have here our pixelated mask for the E. coli, right? This would be the one. So this is at the foreground. And there is two subtle points in segmentation what you want. So in segmentation, you want from this, right? Given the image, you want the mask. And there's two subtle points. You might want for this guy here also to have a 1 if it's what is called here a binary mask. Yeah? So it just tells you what is foreground and what is background, or what is an E. coli pixel and what is a background pixel. Or you might have instance masks. So we want here to have, for example, 2, right? So we want to distinguish that this is a different cell than this. And this is particularly interesting if cells are touching. Yeah? Otherwise, you can say, OK, no problem. Like all my connected components afterwards just get a post-treatment, and they get new labels. But this is a different thing. This is already difficult if they're touching. So, and which they will in this lecture. So let's start. Like in the measurements lecture, we start with super simple, completely oversimplified problems. And this is like an idealized microscopic data. And we wrote just a little drawing library for you. I mean, super simple, even simpler than title graphics. It can just create a canvas, and you can just paint disks on it. And essentially, that's it. And it can show something. So the code, we find the code here. And let's do that. Yeah, our E. coli will be disks on a canvas. Super simple. Yeah? And only, by the way, only black and white for the moment. So we do this. But of course, we want to have a little fun. So we made these two E. coli touch each other, because otherwise it would be too simple. Yeah? And now the task is to segment this, and to instance segment it. So this should be a 1, and this should be a 2, and then there should be a 3. So how can you do this? Right? And there's tons of algorithms that have been proposed, but a repeatedly good algorithm is something that is called watersheding. And we'll show you this picture here. This is the original picture, just with a distance metric applied to it. So what is this distance metric? The distance metric is, I told you before that the background gets for sure the label 0, right? So for in this case, you can. So this is already expecting a black and white image as an input, this distance metric. So we are saying, for example, this gets distance to the background 0 because it's the background. And this pixel gets distance to the background 1 and so on. So for each pixel, you ask, what is the distance to the background? And this gives you a landscape, right? This is very far from the background. This is very far from the background. This is very far from the background, and so on. So this is kind of mountains that go into the sea. And this is our landscape viewed from the top. So you see here a mountain, here a mountain, and here a mountain. And now you can think of like reversing these mountains, like putting them actually as holes and not as mountains. So like just negative distances. And now you start to fill up from the maxima, or the minima in this case. You start to fill up to pour in some water. Let's say you let it rain, and the levels rise. And this is what this watersheding does. So you can think of this hole starts getting filling up, this hole starts getting filling up, and this hole starts getting filling up. And of course, this fills up until it kind of reaches the sea level of 0. But here, what will happen is, at some point, these two water ponds will start to connect. And at this time, you stop it. And you draw this connection line. And if you do this, you get this, right? Like we have filled up this pond, we have filled up this pond and this pond, and they start to touch here at this intersection. And this is the principle. Like we will not go into details, and there is many improvements of this algorithm and also shortcomings. It had been improved, but it's a so prototypical algorithm, I think, that it's worth noting it here. This gives a very nice segmentation of such touching objects, right? Because it actually could separate these two touching objects here. Yeah? Do you see any problems with this approach? I mean, it looked actually too simple, right? We could stop the lecture here, yeah. But there's of course some, since it's the start of the lecture, it's a bit clear, but like there are some problems with this. And one of the problems is noise, right? So we kind of assumed a perfectly thresholded black and white image for this. So it cannot do with any grayscales. We really knew this is the object one, or this is an object, this is the background, this is an object, this is an object. The only problem is they are touching, but otherwise they are objects. Now let's add some noise. Let's say we could not obtain such a perfect black and white image from the beginning, but we had some problems because of some, I don't know, noise going on in the image. Yeah? We put some threshold, let's say, and we obtained something that looks like that. Yeah? I mean, that's perhaps a bit very noisy, but let's get started right into the way. Yeah? And if you do here water shedding, I mean, what will happen? Probably every such pixel will fill up with water, right? So let's try. And here we go. We have tons of cells. This is a cell, this is a cell. All these are small watersheds that are segmented. Yeah. Of course, this is unusable. Yeah. So this is a clear shortcoming of this water shedding algorithm. Yeah. It's very good to separate touching things, but it's very bad to make use of such noise. Yeah? Yeah, very good idea. And this is exactly what is coming next. Yeah, yeah. But very, very good. Thank you very much. Yeah. What we do it a little bit differently, but the concept is exactly the same. What we do here is kind of we let the object, or if you view it as like as a sea with some hills inside there, we let the sea grow a little bit first. Yeah? So we let the background eat up a little bit inside the white things. Yeah? And if you do this, yeah, of course you get smaller objects. And how to compensate this is we reverse the process and we let the object eat up a little bit outside of the background again. Yeah? Of course, you might slightly change the shape of this. Yeah? But if you do this not too much, yeah, then you don't change it too much, actually, the shape. Yeah? And here we see that actually, I mean, this eating up from both sides, that we call it here shrink and grow, yeah? These are very typical functions from the SciPy image processing library. So people do this all the time. So this, of course, there are functions for this. And you see here we tell how many iterations we are eating inside, how many iterations we are growing up outside. Yeah? For some reason, we chose 5. I mean, I think 3 also worked here, just to demonstrate that larger numbers also work. And then we obtain this. So this is not anymore the super perfect circles, yeah? But they are pretty good. Yeah? And then here, of course, this is perfect for water shedding, yeah, right? Here we can do all the way what we wanted, and we get back again our 3 cells. Yeah? Perhaps not pixel perfect, but pretty good. Yeah? Okay. So I would say after this kind of warm-up session, I mean, life will be not so simple, probably, when we look at our E. coli images, right? But we feel kind of warm up. We feel equipped for the real data. So let's look at the real data. And for the real data, what we did is actually we went for you to the lab, and we took some images of our favorite species, the E. coli. And here you see the images. I mean, we could have done better, but this is actually a good training exercise that we didn't do better here. Yeah? And it was quite a not so expensive microscope, yeah, for the image. It's pretty good for the quality of the microscope that we trained this on. Yeah? So what you see here, if you squeeze a bit your eyes, is you see here, and here an E. coli. And here you see some garbage, yeah, that it is not very much defined. So this can be, probably it's something on the lens, yeah, but it could be also some debris. For example, here I think there is a little debris in the medium, but probably this is something on the lens, but we don't care. In all aspects, it's not an E. coli, right? And this is, yeah, by the way, this is a very zoomed in version of a bigger picture, yeah. This is just to get trained on. We cropped that, yeah. So of course it's more sharp if you see that as a whole picture. I think it's not even 5% of the whole picture, so it's very zoomed in. Yeah, just if you're interested, that was done with a 40x lens, yeah. So normally you should do that with a 100x lens, but the setup is much more complicated. This is a super simple setup. I mean, to take this image, it takes like 5 minutes or something. Yeah, yeah, but let's try our algorithm, right? Let's try our water shedding. But to do water shedding, what we have to do first, water shedding takes only a black and white image. It cannot do with grayscale images, huh? at least not in this original version. So we have to threshold it. Yeah? And luckily, if you look at the image, our objects are darker than the background, right? So there is a hope for a right threshold. And of course, you could now plot histograms and look at the peaks and set in the optimum and whatever, but this is an overkill for this simple image. We just played around a little bit with very simple thresholding here. Like this is this here, right? So I think it's originally between 0 and 1, and we scale it to 255, and then we threshold it as 180, yeah? And this is what it gives. So here we really see our 3 E. coli plus a little noise. Yeah? Right? But noise doesn't frighten us, because we have just learned what to do with noise. So we do these steps. Here you have to be careful, right? We are just applying one iteration, because if we are eating up too much, this will go. So we have to be a bit careful here. But you see, like it's already here we are super much into parameter optimization, right? Like we have to set the threshold, we have to set how many do we grow inside or outside, and probably this approach will not work on your next microscopy setup, right? This is something you see here already, and this is very important to notice. Okay, but here it worked. We have our 3 E. coli, and we do water shedding, and here we are. Job done, yeah? So it worked. By the way, how good is this, right? I mean, we at the moment we were to assess whether some segmentation was good. We looked at it, and we said it looks good. That's good. That's fine. But is there some better metric or some objective metric how good a segmentation is? And one of these metrics, or probably the metric to consider, is something which is called IOU. Yeah? So this is if you spell it out, intersection over union. Yeah? And the idea is in principle here. You have here an object. This is our real object. Yeah? This could be the real E. coli. Yeah? And you have a segmentation. Take another color. I had one actually here. So this is our segmentation here. Right? So it's good, but it's a bit off. And what you can ask now is actually to compute the intersection. So the intersection of the two would be this part here, right? That's the intersection. And then what is the union? I mean, the union is like this outside area here. And when I say compute it, I'm referring to the area. Yeah? For example, the number of pixels in this case. And then you are just taking the area of this intersection divided by the area of the union, right? And this is not too difficult to see that, for example, in the case that you have this here and here, right? Like the intersection, in this case, the intersection, the area of the intersection, sorry, would be 0. So actually, your I or U would be 0. Or like in the perfect match, right, you would have IOU of 1. And this gives you kind of like a very nice measure between 0 and 1 of the quality of your segmentation. No? Okay. Yeah, that was just a theory. Like we're not running it here because it's anyway. We will move to more interesting images. Yeah, but this is just a short thing before we go to more challenging data. Okay, how about more challenging data now? So we went to some people who did very nice data sets of microscopy images, and one such that we found very nice is actually a data set by Scher et al. Like what we propose here is like to download this from this website here and just put it in your folder, what we did for you here. And then we are just viewing it. Like it has some directories, it has a train, a validation, like the classical AI things, right? It's done for AI. And we will actually go into AI now very soon. So it's already split up into a train, a test, and a validation data set. And we are just looking at the train data set, the first image that's here. Often these directories are IMG and then a running number and mask underscore running number, yeah. And of course they're matching. So they did the labeling kind of for us. This is very nice for training. So this is an image. And here, I mean, it's already pretty difficult to say what is this is not E. coli, I think. It doesn't look like E. coli, but it's other bacteria. But it's already difficult to say what is here E. coli and what not. And yeah, by the way, here we are looking into masks of but one after the other. Let's apply first our water shedding algorithm. Like we were quite proud of the water shedding algorithm, right? Let's apply that to this image. So this is the best threshold we find. I mean, you have to trust me. I played around a bit. Doesn't get any much better. Here is the water shedding segmentation of it. Does this look good? Like that's our bacteria segmentation. It's telling me here's a bacteria, here's the next one, here's the next one. Here's the next one, here's another, here's another, and so on. I mean, it doesn't look good. And it doesn't get better. I mean, you can really play around, but it doesn't get better. So we have to get to more mighty tools. And we go to the holy AI here, of course. Yeah? So let's look at deep learning. Yeah? And so for deep learning, there's tons of deep learning libraries that can do segmentation. What we choose here is a library that is perhaps not the very fastest one, but that is very didactic and very universal. So it's not very specific to the task. It's a general segmentation library. So people use this, for example, to segment cars in scenes or pedestrians or houses or whatnot, cats. It comes like we combined it with we're using it with a light version of PyTorch, because it's also we're not interested in getting the last thing out of it, so we want to be quick and slim code, and this is why we chose that. And you will see this in a second. So the segmentation library is called Segmentation Models PyTorch, and we will be using other libraries that are quite interesting. So albuminations, it's called. So this is actually for image augmentation. So our data set is not too big. I think it's about 15 or something images that have been hand segmented. So that's not very good for AI. You need much more. And such libraries help a lot with that. They can rotate, put noise, flip, crop, and whatever to have tons and tons and tons of images from a few images only. And then we are using this light version of PyTorch that helps us speed up simple processes that are a bit coding heavy in PyTorch. This is only for didactic reasons, essentially. Here we have our directories. Perhaps at this point, a big thanks to to the segmentation models authors, because they have put a very nice tutorial, and we are like, the tutorial is for cars, and we are kind of adopting it for equality here. And you will see that it works very nicely. Yeah, this is really just some image processing is a lot about grabbing the right files, putting in the right order, transforming them, and so on. I let you read the code here. You will get very acquainted afterwards with list-dir, make-dirs, and so on and so on. Yeah? But it's very practical things. Yeah? So this is our images. So actually we have 22 images here, as you see in the training data. That's not too much for deep learning. We have only a single class, right? The only class we have actually is a cell, like an equality. Everything else is just a background. Huh? This is the data loader, and perhaps the most interesting thing of this data loader is it just pulls from the directory of images. Yeah? But the most interesting thing of this data loader is it's applying augmentations. Yeah? So you can set it when you create this data loader, you can set the augmentation, and it's just going to apply. If it's set, it's just going to apply the augmentation, right? And of course, it has to always apply to the image and the mask, yeah? Because they should be, if you rotate that image, you should rotate the mask in the same way, right? And then it's just returning the augmented image mask. Yeah, and here you see, these are the little traps that cost you some hours. Yeah? Here you have to transpose it because you're going from NumPy to PyTorch. And PyTorch really wants this thing, yeah? Like be super careful about that things. They cost a lot. Yeah, this is just to visualize. This is nothing big. And here we are visualizing our data set. And we are just looking into the first image and the cell mask of the real data. And this is how they look like. Yeah? It's very well hand augmented, of course. And of course, yeah, what you see already, these objects are not bacteria, right? As you see here. And yeah, another thing we see here, it's this is not instance segmented, but it's like we just have a 1 and a 0. It's a cell or it's nothing. Yeah? Okay. This is quite fun because here you have to think of what are meaningful augmentations. Right? What are meaningful things you might want to do to your pictures? And this depends a lot on your application. You will do completely different things, for example, if I take a picture of this classroom, I might take completely different augmentations than I'm using here. For example, one thing I can tell you, we are not using any how is it called, like perspective augmentations, right? It doesn't make sense. I mean, for example, if I take the picture from here, I can compute it mathematically that it looks like I took it from here, right? But this really doesn't make sense for a microscope. You're not going to sometimes microscope in this way and sometimes from this side. Yeah? This is an augmentation you will drop here. And some others, so this is very particular already to ours. Yeah? Classical ones is flipping. And this is a very nice library. You can say, for example, whenever you do an augmentation, flip it with a probability of 0.5, yeah? Or do an affine transformation within these bounds. It's choosing this randomly. It's randomly rotating between bounds 44 and 45. And then pad if it's needed, or do a crop with probability 1, so always do a crop, and so on and so on. Yeah? So this is a super powerful library. You can even say, do one of these things here, and so on. So it's a very nice pipeline for augmentations. And that's after augmentation. So augmenting the same image, we get, for example, this and this instances, right? So you see here it has been turned a bit, made a bit darker. Here it has been cropped, I think, and so on. Yeah, like we get tons and tons of images suddenly. Yeah? Yeah, this is just creating the data sets with these augmentations. And finally, creating the model. You see, it's the forward step in this neural network is super simple. It's just doing a normalization and then calling the neural network. And there is tons of logging steps that I will not go into here. And then we are just running. We are running this. Where is it? Yeah, we are training it, of course. Yeah, here we have in the metrics we have here actually this metric that you already know. So for example, here we have a per image IOU, and then we have a dataset IOU, so they are a bit different. Like if this is computed per image or for the complete dataset. And after lots of training, like the training was done on this MacBook Pro, I would say it was done for 25 minutes, so it's actually reasonable. Yeah? So this has a GPU, but it's not a super strong GPU, but it's okay. Yeah. We get actually this on the validation dataset. Yeah? That looks pretty good actually. Yeah. I mean, it's a bit flu here and there. There is some pixels that are not here, but otherwise it's pretty good. I mean, this is only 25 minutes. I mean, this can be much more improved, of course. Yeah? But it's just for running it on a laptop. It's pretty good. Yeah? And this has never seen any kind of cells probably. Yeah, right? This was trained probably on yeah. It can be used on cats and dogs and whatever. Yeah? So it's a very standard thing. It has nothing to do with microbiology. Yeah, probably. Okay. And the images are quite challenging, right? Like you see these things here. It's probably from a microfluidic device. It does this. So it's pretty good. There is a but. Yeah? We got this data from a publication, and now we are applying it to our nice microscopic image. Right? You might remember that. So it was actually a crop that we showed, and here is another crop, but you see here again the E. coli, if you squeeze your eyes, yeah? and we let it run on it, and it predicts this. There is no cells on it. So it's clearly not able to generalize to completely different scenes like this. Yeah? Okay, let me just give me a second. Yeah. Completely. I mean, if you see the type of what a bacteria looks in this here is completely different to ours. Yeah. I mean, this looks very much like phase contrast images. You have this halo, I don't know how to pronounce it in English, around them that are typical from phase contrast images. It's much cleaner. It's probably a very good microscope. Yeah. Something around, I guess, 100x. Yeah. Mm-hmm. Yeah. No, it was only. Yeah. This is what I mentioned was with it's done with a 40x microscope. Yeah. And it's a super simple setup. Like again, like to take this image, it was 5 minutes. You put the slide under, you snapshot, and that's it. You can do much better with oil immersion microscope and so on and so on. Yeah. But this was not the purpose. Like we wanted to have something that it's with super simple setup, like a Raspberry Pi camera. I don't know, costs 20 euros. Raspberry Pi costs perhaps 100 euros. Yeah. Put it together in 5 minutes and make a picture. That's not the. Yeah. And clearly it looks completely different for such images. Yeah. Yeah. And it just didn't generalize to that. Yeah? Perhaps it's not very surprising, but I just want to show you that's kind of the limitation. We should not expect magic from this to happen, the generalizability. I didn't try it on a cropped image. I think I tried it once. I cannot 100% promise you, but I think I tried it once and it showed the same problem. I think it was not a problem of that it was zoomed in or zoomed out or something. I guess it's really the difference between the background and the foreground in our image types, it's very fluid and very slight, and the others it's a very sharp with this halos around it. It's a different type of. It's a different type of microscopy. Here we are using bright field microscopy, and they are using phase contrast. So it would be very good to generalize, but it just doesn't do this in this case. Yeah. The last thing I want to show you, of course, what we could do now is, right, we could run it on our images, right? And nothing prevents us from this. And actually, we are going to do this, but we are going to do this with something that is particularly done, like it's a framework particularly done to segment cells. It's also a deep learning AI software, but it's really done, it's specific to cells, while the other is really any kind of object in images. And one of these dedicated cell segmenters, deep learning-based cell segmenters, is a cell segmenter that's called CellPose. Right? Yeah, again, I think it's a very didactic one, works very well, and you see its training is a bit more difficult because it's much more training intensive, but it's a really nice library. So I will not walk you too much into this code because such image processing code is always very file heavy or transformation heavy, right? Like we have to get the E. coli file, for example, from our microscope. We have to read it with the right library. You have to be here careful with what image read you read from which library to know how it's normalized or not normalized. There's all these little caveats. We are kind of cropping it. Something that you see a lot in deep learning image processing is there are always multiples of 32. This has to be to do with the architectures they're using. Some small details. Or for example, here once you did this transformation, here we crop it, right? We have to cast it to a different bit width and so on. If you forget this, something bad happens. So it's very tricky, these things. The difficulty is a bit in the detail, right? Yeah. So we are going to run a pre-trained model on this, right? And this is what we get. Is this good or bad? What would you say? It's good and bad, I would say, right? It's very bad because it didn't get a single of the E. coli, but not a single one. But it's pretty good because it got all the circles. Yeah. So it's thinking these are cells. Probably it was trained it's again, it's a cell segmenter. Like it can, for example, this could be yeast cells or they could be any kind of round cells, right? So it does a pretty good job, but not what we want. So that means actually we have to train it, right? And this is what we did here. Again, lots of image putting around. Perhaps I show you the interesting parts of the code. Yeah. What are the interesting parts of the code? So first, again, we don't have a lot of images. Yeah. Why? We have actually a lot of images because the images we got and they are checked in, they are from a long video. Yeah? We have, I think, a 1 hour video of such E. coli flowing around, so we have tons of images. The problem is we don't have many manually segmented images. Because why should we? So what we did for you is we hand segmented for you 2 images. Yeah? But already 2 images takes a bit time. Yeah? It's not too long. I would say per image you take 5 minutes. Yeah? So it would be 10 minutes. But we wanted to demonstrate you that you don't need to segment by hand 100 images. Yeah? You can do actually with 2 images if you use it cleverly. Yeah? And so we hand segmented 2 images with a very nice software. That is, wait, I want to kind of show you the software here. It's this software here, right? And this allows you to, you can call it from Python, and you can add an image. You can add a label that you, for example, you say, please segment me something which is called E. coli, right? And then you can just block it, and it pops up a GUI. And it tells me, please do the segmentation for me, right? And then you have to really, with a pencil, you have to paint around the equals in different colors. Don't go here because of the time, but essentially, and then when you close it, this stops blocking, and then you just read out the labels and save it as an image, right? So this keeps popping up in the loop, and then you can call it, for example, you can call it like this. You can call it annotate cell image 0. It's just commented out that if you run the whole sheet, you're not always popping up the segment, the hand segment. But if you call that, that will actually that will allow you to segment image 0, and here we segmented image 10 for you. Because if you be very careful with the segmentation, if you would have segmented image 0 and 1, they are almost identical. They are from a video, right? So you might bias your training a lot on the same bacteria. 10 is far enough that you have new E. coli on it. Okay. And this is how it looks after segmenting. This is the training data set. Yeah, and of course, like you see, this is already a crop because we used actually the transformation library. I mean, we segmented 2 images. This is clearly not a good idea for deep learning. So we have tons and tons of generated images from rotating, flipping, cropping, and doing fun stuff. This is how we got, for example, this image here. And of course, the segmentation this is now an instance segmentation. So this is really a different label than this. This might be, background is label 0. Let's say label 1, 2, 3, 4, 5. Right? And now we have this. We are ready to learn. So we are creating the cell pose model like with this. This is now it's super simple. Yeah? We create this. We might take just a little glimpse at the neural network, just for interest. Yeah? And what we see is a transformer. Yeah? Surprise, surprise. Yeah? Works very well, as we see. And yeah, we train. And this is something we made a little shortcut for you. Actually, when I trained this yesterday or the day before yesterday, on the MacBook Pro, I just stopped it at some point because I think I was reaching something like 10 epochs after 1 hour, which didn't move anything. Yeah? So I just stopped it. And we fortunately have an AI server in our lab. So I went to this AI server, ran it, and it was done in, I don't know, it was done in 10 minutes with 100 epochs. So it has a nice GPU from NVIDIA, and it's a huge difference. Both were run on GPU, but the Mac GPU just cannot even closely compete to some AI GPU. Yeah, and yeah, exactly. And to the shortcut, if you don't have this very nice AI server at home, and you still want to try that out, we posted something on Hugging Face, a pre-trained model. So you can just download that. Okay, here's the training. Training. And this is something you should not be surprised of because, wait, let me just check what I... Give me a second, just.
There is a question from Zoom.
Okay, yeah, sure, yeah. Yeah, definitely. And several of our students do this. So they use Google Colab, and they have access to, you get GPU time. It's definitely possible. I just didn't try it out here with this notebook. So please you have to be a bit careful how to, I think it's always a bit tricky to upload your data into the Google Drive and then to get it inside, because it comes with a bit of data, not too much, but you might check that out. And I think the model alone is around 1 gigabyte. Yeah, so please check out. We didn't test it, but it should be possible, totally. Yeah, no. You mean the hand segmentation, whether this is biased, like the initial ground data? Okay, yeah, 100%. And actually, it's a very good point because, for example, when I was first running this notebook, I did an initial segmentation very sloppily. I just put with the pen, like in 1 second, lines for the E. coli. And actually, when I looked at the crops here, yeah, they were a bit off. So the ground truth masks didn't really match. Yeah? And this definitely influenced the training. So what I did is, like, in the second pass, I spent a bit more time. But you don't have to be pixel accurate. For example, I mean, you see here there's E. coli. Don't overdo that. Yeah, whether you paint a bit outside or a bit inside, it's not so important. Yeah? So I didn't take a very long time to. I would say probably I segmented a complete image with 15 bacteria in 4 minutes or something. So don't overdo it. Mm-hmm. But definitely it creates. If you don't have a good ground truth, it will not converge very nicely. Yeah. Thanks for the questions. Yeah? Thank you. So this is what's shown as the training data, right? And this is this is the training data. This is the input of the training data. So this is the ground truth, right? And of course it matches, because if I didn't do a mistake, it should match. And this is a very nice test that I suggest to do for you. Whenever you train something, let the neural network run on the training data again, or an instance of the training data. Right? This is completely against what you learned from the last 2 lectures, right? But it's just to test if your training loop kind of worked. Yeah? It doesn't look into generalization. Of course, you should afterwards look at into the test data and the validation data. But it's just, did this learn anything? Yeah? Because if you get any of these transformations or bit depths or whatever wrong, this could be, for example, a completely black mask. So then you know you don't even have to look any further. If you didn't learn the training data, right, and you presented the exactly same image you trained it on, there is no hope it does anything. Yeah? So this is just this very simple test here. So this is not kind of quality, it's just an assertion. It did learn something. Yeah? Okay, but now let's do the real test. So we are calling it on the image number 25, which we didn't show it yet. Has never seen it. Remember, we have trained it on 2 images, right? That's very few for deep learning. And we now give it a completely new image. So we crop it, just to see it in more detail. But you could give it also the complete image, so that would also work. And that's the result. I would say it's pretty good, yeah? Like there is this one, and there's this one and this one. Yeah? So that's deep learning on 2 images. Yeah? And it's not to tell you that you should deep learn everything on 2 images. Yeah? This is not the takeaway message from today. Yeah? It is just, it's impressively good if you do the right choices and the right augmentations and the right pre-trained models, you can get impressively good results. Yeah? And this brings me to the end. Of course, if this would be a project, right, we would have just started now. Right? I mean, we got very promising results, really works awesome. But there is a but, of course. We have tested this on a crop. Right? I mean, I have tested it also on other images, looks good, but now it's to do this systematically. Right? You should really validate performance, for example, with this measure, IOU. Yeah? You should run it on a separate validation set, whether this does really what you want. You could, for example, count, right? You could count the cells here. It's 2, and you can annotate the validation data set and count this and say, does it get the right count? Probably you have to change a lot of the learning parameters, right? I mean, they are just standard learning parameters we used here. You have to tune this probably to get better results. And finally, yeah. And then probably, perhaps even you have to change the architecture, right? We just use the standard transformer that comes with this. You might want to change the architecture even. Right? Okay, but I think it has helped you to get a small glimpse into what to do with microscopy, from super simple classical algorithms like watersheding, pixel reductions, to more complex segmentation software, general software, to even more specialized cell segmenters, and to training them. And in the problem session, you will actually train it. We're discussing an interesting perspective of if you have few data in the problem session. I just show you this perhaps for a second because I think it might be interesting from the concept. Don't worry, I'm not going over details. But what we're discussing is a way to get simulated data to train something, right? Like what we have looked into, let's say we have only 2 images, and we want to deep learn on them by augmenting the images. But sometimes you have to start with 0 images because you don't even have the setup yet. And then a promising approach is actually to generate synthetic data. Like that looks pretty much like a microscopy image, right? And this is purely synthetic data. Yeah? This experiment was never run. And this is like generated with the help of some very nice library. Yeah? Okay, then that's the end of the lecture. Are there some questions?
There is a question.
Yeah, yes, please. Yeah. Yeah, definitely. So ImageJ is a great product. I mean, and it's very. I think it's so. We didn't present it here because it's a bit our philosophy to have everything completely run in Python. Everything is a code, right? That's the reason we wanted to show you how you can kind of code everything from the bottom to the top. But ImageJ is a very good tool, I think, to prototype, to look into such pipelines. Because the problem with kind of the downside of the approach I showed you here is like you have to code everything out. Yeah, if you decide, for example, to use this and this segmenter, you have to kind of code it. Yeah, or you have to decide another segmenter, you have to kind of code it. In ImageJ, the nice thing is you can choose from a large variety of tools, of plugins, and you can just try out. You can just say, what would this segmenter give? No, what does this watersheding give? Oh, could I add a Gaussian blur first and then do another edge detection and then only watersheding or whatever? Yeah? You can very quickly prototype. Yeah? So I think it's super nice for getting a feeling for different methods for prototyping. It comes with the problems of a GUI-based method, right? Like of not having code as a first principle run through your process. Huh? But there's nothing wrong with ImageJ, of course. Huh? And I think there is this successor library which is Fuji, or it's called. You might want to look also into this. Huh? Yeah, they are very good tools, definitely. Yeah. Did this answer your question? Or? Oh, okay. Okay. Thanks. Thanks. Okay. If there is no more questions, I think we can stop here and wish you much fun with the problem session. Yeah?